Welcome dear students. In this video we are going to learn about Rapoport-Leubering shunt. This shunt or the cycle is actually related with glycolysis. So let me draw the glycolysis. Glycolysis starts with glucose, then the glucose is converted into glucose 6-phosphate, then glucose 6-phosphate converted into fructose 6-phosphate, then the fructose 6-phosphate is converted into fructose 1,6-bisphosphate. I am not showing the reversible or irreversible step, not actually will write the name of the enzyme. I will show where this Rapoport-Leubering shunt is actually related with, where the Rapoport-Leubering shunt is related with the glycolysis. So from the fructose 1,6-bisphosphate it gets splitted and becomes DHAP and the glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate. That means these are actually molecules of triose, means it contains 3 carbon molecules, because we know that glucose is 6 carbon molecules. So these are the 3 carbon molecules and this is another 3 carbon molecules. Now this dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate and makes the 2-glyceraldehyde 3-phosphate. Now from this glyceraldehyde 3-phosphate 1,3-bisphosphoglycerate are produced. From the 1,3-bisphosphoglycerate 3-phosphoglycerate are produced. From the 3-phosphoglycerate 2-phosphoglycerate are produced, and 2-phosphoglycerate phosphoenol pyruvate or pyruvic acid are produced, and ultimately pyruvate is the ultimate product, pyruvate of the glycolysis. Now this Rapoport-Leubering shunt is actually related in these portions, in this section. In this section the Rapoport-Leubering shunt is actually related. And how? I will show you. The enzyme that actually convert the 1,3-bisphosphoglycerate to 3-phosphoglycerate are called phosphoglycerate kinase. Phosphoglycerate kinase actually convert ADP to ATP. To ADP actually converted into 2-ATP, and this type of production of ATP is called oxidative substrate level phosphorylations. It is totally different from oxidative phosphorylations. Now this kinase enzyme makes this 3-phosphoglycerate actually remove one phosphate from the carbon 1 to make ATP. Now the Rapoport-Leubering shunt is actually converge or actually bypass this step, and what happens? From the 1,3-bisphosphoglycerate a product is actually generated we call 2,3-bisphosphoglycerate. There is a difference. So the Rapoport-Leubering shunt, these actually are 2 Austrian biochemist scientists. They discovered this pathway. Specifically, you remember this pathway or shunt is actually occur in erythrocyte, that means RBC. So this take place in the RBC, and there is a reason I will tell you. So 2,3-bisphosphoglycerate are produced, so it actually bypass the main flow of this glycolysis. Now this 2,3-bisphosphoglycerate is actually converted back into 3-phosphoglycerate. Now 2 enzymes actually take part in this Rapoport-Leubering shunt. The enzyme name is, so this enzyme is actually action taking part in these sections. So I am writing here the enzyme name is the phosphoglycerate, or you can say it could be 2,3-bisphosphoglycerate mutage. So you can simply remember only phosphoglycerate mutage. We know that mutage enzyme can transfer phosphate group from one carbon to the next carbon or another carbon. Like here that there are 3 carbon, because this is a 3 carbon product I have mentioned here, 3 carbon. This is also 3 carbon product. So there should be 3 carbon, and from in the first carbon there is a phosphate, PO3 minus 2, and the third carbon there is a phosphate, PO3 minus 2. Now this enzyme, this mutage, bypass this pathway because it actually convert this 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate. What happens here? Here the mutage enzyme actually remove or actually transfer this phosphate to the second position. Let suppose this is the first, second and third carbon. So transfer this first phosphate to the second carbon, sorry. So it make 2,3-bisphosphoglycerate. Now this 2,3-bisphosphoglycerate is very important. 2,3-BPG is important. Now this 2,3-BPG then converted back into 3-phosphoglycerate, and the glycolysis pathway occurs. And the enzyme that is responsible for this transformation is called phosphoglycerate phosphatase. Why it is phosphatase? Because this enzyme removes the second phosphate group from the second carbon, so that this 2,3-bisphosphoglycerate can become 3-phosphoglycerate. This is the Rapoport-Leubering shunt. They have found, particularly in the RBC, this step is actually bypassed and it make 2,3-BPG from the 1,3-BPG, and ultimately it actually becomes 3-phosphoglycerate by help of this two enzyme. Now what is the role of this 2,3-BPG? Yeah, there is an important role in RBC of this 2,3-BPG because this is actually negatively charged molecules, and it actually helps in the releasing or unloading. It actually helps in the unloading of oxygen from the hemoglobin. This is the reason. Unloading oxygen from the hemoglobin to the required tissue site. So this is the Rapoport-Leubering shunt. Hope this video will help you. Thank you for watching, and always keep learning.